Hello friends, welcome back to my channel, and today in this video we are going to study about receptor-ligand interactions. As you can see in this diagram, a receptor is a membrane-bound molecule. Now this receptor will bind to a ligand. Now sometimes the ligand can be secreted by any nearby cells, or sometimes this ligand have to travel a long distance to bind to a receptor. So what is a ligand? Ligand may be a soluble molecule, or it may be a peptide, carbohydrate or lipid presented on the surface of a cell. Now if we study nervous system, then any neurotransmitter can work as a ligand. If we are studying endocrine system, then any hormones can be a ligand. And because now we are studying the immune system, so antigens, cytokines, these are the ligand molecules that bind with the receptor. Once the ligand bind with the receptor, what will happen? A conformational change will occur at the cytoplasmic domain. This is the cytoplasmic domain of the receptor. So a conformational change will occur in the cytoplasmic domain of the receptor, or sometimes it's a molecular change due to which the intracellular signaling events will start. And as a result, some gene will express to respond to the ligand present in the medium. So depending upon the ligand, the intracellular events will pass and will express some genes to release any compound or to behave in response to that ligand. If it is an antigen, it may release some cytokines, or this signaling can cause the proliferation of any immune cells due to which it becomes more reactive against the antigen. If it is a cytokine, then it may undergo the changes like proliferation, differentiation of the cell, or will release some other cytokines to interact with the antigens. So depending upon the ligand, the cell has to respond. Now the cell can only respond when the signal gets transmitted up to the DNA or the gene level, and the gene will express accordingly. That particular cell will respond to the ligand. Now what are the bonds involved in receptor ligand binding? So receptor ligand binding, it should not be that strong that ligand cannot get separated from the receptor. So it should not be covalent binding. So the receptor and ligand binding should not be covalent in nature. So what other interaction? The non-covalent binding. Under non-covalent binding, it can be hydrogen bond, ionic bond, hydrophobic interaction, van der Waals interactions, or ionic bond. Again, so ionic bond means when any positive or negative charges are present nearby. Hydrophobic interaction means when any hydrophobic molecules accumulate or gather and they interact, that is a hydrophobic interaction. When two molecules are nearby, so because of that, the force comes into place, the van der Waals interaction. Hydrogen bond means either the H is present with O or with N. This can form hydrogen bond with another molecule. So like this, with all the non-covalent interactions, the receptor bind the ligand with sufficient binding energy. Now, what is the strength of receptor ligand interaction? R, that is receptor plus ligand, is reversibly equal to receptor ligand complex. The forward reaction is given by K1, the reverse reaction is given by K minus 1. Ka, that is association constant, will be the concentration of receptor ligand complex divided by the concentration of receptor into concentration of ligand. So because it is an association constant, so more the value of Ka, more will be the association or the interaction or the affinity. The opposite is true with the dissociation constant. So dissociation constant means individual concentration of receptor and ligand divided by concentration of receptor ligand complex. Because it is a dissociation constant, so lower the value of Kd, more will be the receptor ligand concentration or affinity or binding. Now the Kd value of antigen antibody interaction at the beginning of the immune response is normally what is the order 10 to the power minus 4 to 10 to the power minus 5 at the beginning of the immune response. Now what should be the value of antigen antibody interaction at the late of the immune system? So in the late of the immune response, the bonding will become more tight, the affinity will increase. So you have to tell me whether the value will increase this way or this is correct. So you have to tell me which direction is correct. So towards the late of the immune response, the affinity should increase. So now the interaction can be multivalent. Multivalent means more than one ligand binding site per molecule. Now the multivalent or multiple concurrent receptor ligand interaction, it increases the strength of the binding between two surface. So this is antigen and this is antibody. So if one binding is there and simultaneously with the same antigen two binding is there, so definitely this will be the strong. But this strong binding won't be double. If I show one arrow for this, I cannot show two arrow for this. So the interaction will be definitely stronger than this, but it won't be double. Why it won't be double? Because when it is binding with two sides, then the binding will at one side will somehow strain the molecule, because as you can see here in this diagram, with one it is fitting properly, with one it has to bend a little. So that means binding at one location, it will strain the binding with at another location. So the binding is definitely will more compared to binding with one side, but it won't be double, because binding at one side will strain the binding at another side. And when more than one binding site is involved, it is less likely that all of the receptor will simultaneously be in off state. So what is happen? It is in the on off state. What do we mean by on off state? Suppose it is binding with two ligand. So on off means it can happen that at some particular time it is binding with this site, at some particular time it is binding with the other side. So it is like on and off. It cannot be like off completely or on completely. It is be in a state of on and off. So that is known as multivalent binding. Now for multivalent binding, we use a term that is called avidity. Avidity is used to describe the overall strength of the collective binding interactions that occur during multivalent binding. Now, what is the molecular logic of immune system? As we already learned, whenever any antigen presenting cells interact with naive T cells, this naive TH cells, because it is CD4, so it is a TH cell, it can release a variety of cytokines. Now, what cytokines it will release? Finally, affect which molecule it will activate, either B cell or macrophages or TC cells. So depending upon which cytokine this CD4 plus cell is releasing. Now the receptors for some growth factors and cytokines, these are expressed only on an as needed basis. It is not like that these cytokines will be releasing in our body all the time. It will only get released when needed. Now the secretion of cytokines into exactly the location, because one cell which is secreting cytokines is a tiny molecules. The other cell should have the receptor to accept it. Now, if these two cells are very far from each other, this cytokine will disperse. This cytokine will get dispersed or distributed, due to which very low number will reach to the target cell. So because of that, the two cells, one which is releasing the cytokine and one which has the receptor for the cytokine, these are mostly closely packed, and the cytokines are directly released towards that acceptor cell. And because of which what happens? Microtubule organizing center redistribution. What do we mean by microtubule organizing center? So if this is the cell, suppose this cell has to release the cytokine. Suppose this is the storehouse of the cytokine. Now the target cell is present here. So the microtubule organizing center, what it will do? It helps to move this vesicle towards this side, so that it will release the cytokines directly in front of the target cell. So that is the function of microtubule organizing center. So that because cytokines are the soluble small molecules, it can easily get dispersed and distributed if the target cell is very far away. So in that case, the target cell won't get sufficient cytokines to become activated. So due to which the cells that release cytokines and the target cell for it should be in close proximity, and the cytokines are released towards the direction of the target site only. If the target site is present, suppose here, so the microtubules will organize this vesicle here and it will release the cytokines. Okay, so that's all for this video. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.